Welcome everyone to this week's edition of the Extracellular Vesicle Club. This is actually the year anniversary of when we started this virtual event. Previously, we had been holding a journal club at Johns Hopkins University just once a month. And when the pandemic came along, I started looking for ways to make sure that we could continue that journal club for the local community. But we eventually decided to just make this open for anybody who wanted to participate. And after a very disturbing Zoom bombing incident in the very beginning, we decided to take it to an email-based distribution of a weekly link. And that's worked out pretty well over the last year. I'm not wanting to dwell in the past today, but rather look towards the future and what EV Club will be doing in the next year. If you have any suggestions, or if anybody would like to moderate an event or present at an event, please contact me and let me know. We're also taking suggestions for papers or for topics for future events. So it's my pleasure to introduce Zach Troyer from Case Western University. Zach is going to be telling us about some very interesting work that he's been working on over the last year to address COVID-19 and looking at how extracellular vesicles that carry viral components might be able to affect the virus. So Zach, I'm really very appreciative that you decided to share this work with us today. So Zach, it's over to you. I'll hand the screen sharing over to you.
Thanks, Dr. Whitwer. So I'd just like to thank everyone at the EV Journal Club and Dr. Whitwer for allowing me to present today for today's Journal Club. And just as a reminder, as Dr. Whitwer mentioned, this is unpublished data, so please do not screenshot or share this. My talk and our paper is titled Extracellular Vesicles Carry SARS-CoV-2 Spike Protein and Serve as Decoys for Neutralizing Antibodies. So to give a little bit of background rationale for this work, I wanted to talk a little bit about viruses and neutralizing antibodies. So neutralizing antibodies are the adaptive immune response typically against viral infection. They're antibodies that typically bind to viral envelope glycoproteins and prevent them from infecting cells, hence the term neutralization. Now, these viral envelope glycoproteins are the proteins that viruses use to both bind to cellular receptors and to perform membrane fusion and allow the viral cargo to be released into the infected cell. Hence why the antibodies typically target them. If you can block that interaction between the viral envelope protein and the receptor, then you can block infection. Now, viruses have evolved different methods to escape these neutralizing antibodies, particularly something I like to call decoys. And here I've just listed two papers that discuss such things. This first one is in the context of HIV, where there was a finding that immature HIV-1 particles, which are basically HIV that are not properly processed by the HIV-1 protease, those immature particles, while being non-infectious, they strongly bind neutralizing antibodies. And this has the effect of allowing infectious HIV particles to escape detection, because these immature particles can, in a sense, take the bullet for the infectious virions. In a similar sense, this has been found to happen in hepatitis B, where these subviral HBV particles are released from infected cells. These subviral particles are about half the size of the normal virions, but they do carry the envelope protein of hepatitis B, and they can bind to these neutralizing antibodies, allowing the infectious particles to escape detection and increasing infection. So just kind of as a graphical representation of what I'm talking about, you can see we have a neutralizing antibody here, our infectious viral particle, and these either subviral or defective viral particles that block the action of the neutralizing antibody by serving as decoy targets and allow the infectious virion to infect the cell, which leads to effects of increased infection and decreased neutralization. Now, to bring EVs into the picture, how do we exactly relate this to EVs? Well, it's well known that EVs and viruses have substantial levels of overlap. And there's a really good review here where I've taken this picture from that just kind of explains the connection between viruses and EVs. They share a lot of different egress pathways. They're very similar in size, and they can also carry cellular. They share similar cellular proteins. And instead of thinking about a virally infected cell only releasing viruses and EVs, it's more likely that there's a gradient of particles being released between viruses and EVs, which is kind of represented here. And the ones that we're very interested in are these virus-induced EVs, which are mostly resemble normal host EVs, but they might carry one or two viral proteins on their surface. And specifically, I'm interested in when they carry viral envelope glycoproteins on their surface, because as I mentioned before, these viral envelope proteins are the targets of neutralizing antibodies. And as I mentioned, EVs can carry these viral envelope glycoproteins. I've listed another 4 studies here that have really shown this very well, where we've seen EVs carrying proteins from VSVG, from the influenza virus, from HIV, which is the GP120 protein, and from human CMV. So there's plenty of papers out there suggesting that extracellular vesicles can incorporate these viral envelope glycoproteins. But what about SARS-CoV-2? So SARS-CoV-2 obviously is very relevant given today's pandemic and the current pandemic that's ongoing. It consists of, in terms of its virion, it consists of 4 structural proteins, but the one that I'm very interested in is this spike protein, which is SARS-CoV-2's viral envelope glycoprotein. It consists of 2 subunits, S1 and S2. S1 is responsible for binding to ACE2 and internalization into the cell, and S2 is responsible for the membrane fusion steps of viral entry. And our question was, can extracellular vesicles carry SARS-CoV-2 spike protein? And furthermore, if that is the case, can these extracellular vesicles with the spike protein serve as decoys for neutralizing antibodies in a similar sense to how subviral particles and non-infectious viral particles can? Given that they have relation to, or they're very similar to viruses in some senses. So just kind of as another graphical abstract of what I'm talking about, you can imagine a situation where we have infectious SARS-CoV-2 virions and an ACE2 expressing cell. That cell, of course, can be infected. Now, in the context of an anti-spike neutralizing antibody, the antibody will bind to these virions and prevent infection, hence the term neutralization. If there are spike-containing extracellular vesicles floating around, these EVs could perhaps serve as decoy targets for these neutralizing antibodies, allowing infectious SARS-CoV-2 virions to escape the immune system and infect cells, even in the context of an immune response. So in order to look at this phenomenon and see if this is happening, we produced EVs by transfecting HEC293 T cells with a codon-optimized SARS-CoV-2 spike protein. We also, of course, had a mock transfected control to have the just normal HEC293T derived EVs. We take that conditioned media off of these HEC293Ts, and we initially run it through a 0.45 micron filter to remove cells and cell debris. We then pool that conditioned media, which we now call clarified conditioned media, and we use a technique called tangential flow filtration to concentrate and purify our extracellular vesicle population. So with these purified EVs, we then do a variety of different characterizations, such as multifluidic resistive pulse sensing, nanoscale flow cytometry, transmission electron microscopy, and different proteomics assays. And then we follow that up by performing functional assays, looking to see if extracellular vesicles can serve as decoys for these anti-spike neutralizing antibodies. So for some of our initial experiments, we worked with a version of the SARS-CoV-2 spike protein with an EGFP tag, and we just wanted to make sure that we could visualize spike protein expression, make sure we could express the spike protein in our cells. And that's what you see here. You can see that our spike EGFP is nicely expressed in the HEC293Ts, and interestingly, the localizations does not seem to be at the plasma membrane, but rather it seems to be localized towards the center of the cell, perhaps near the ERGIC, the ER-Golgi intermediate compartment, which has been reported for SARS-CoV-2 spike. But it's a little hard to say with just these fluorescent images, but you can clearly see that it's not seemingly at the plasma membrane. Now, moving forward with this spike EGFP, we wanted to determine if extracellular vesicles or particles released by these HEC293 T cells were incorporating this spike EGFP protein. So for that, we utilized nanoscale flow cytometry and fluorescence triggering to be able to detect events that were normally too small to detect by forward and side scatter. And in here, what you can see is we took the EVs from mock transfected cells, and we don't really, we don't see a lot of events above the noise. However, when we overexpress this spike EGFP protein in our HEC293 T cells, we see a massive increase in the number of detected events, which suggests to us that there are particles being released from these cells that do contain the spike EGFP, although at this point we can't necessarily call them extracellular vesicles. Now, to determine if these were extracellular vesicles that were being released, we co-expressed fluorescent versions of the common tetraspanins CD9 and CD81. And what you're seeing here in this co-expression plot is that there are populations of CD9 positive extracellular vesicles that also carry the SARS-CoV-2 spike EGFP protein, although the majority of the detected EGFP incorporating particles were not CD9 positive EVs. So it seems like the CD9 fraction of these spike EGFP EVs is low. About 8% of these spike EGFP events are CD9 positive. Now, performing the same experiment, but with CD81, we see something quite different. We see that there are a lot of CD81 positive vesicles being released from these HEC293 T cells, but moreover, we see that about 80% of our spike EGFP incorporating particles appear to be CD81 positive EVs. And of course, some of this 80% could also include this 9% of detected CD9 positive EVs, because we didn't express both these tetraspanins at the same time. But it does appear that a majority of the spike EGFP positive events are CD81 positive. So now that we've demonstrated we can that the spike protein, at least with the EGFP tag, is incorporated into extracellular vesicles, we wanted to do further characterization with the wild type spike the wild type spike protein. So here on the left, you can see we've done multifluidic resistive pulse sensing, which is a method used to get particle counts and size distributions of extracellular vesicle preps. So we used an instrument called the Spectrodyne NCS1 for this. And you can see that with both our spike positive EVs, which are EVs released from cells transfected with the SARS-CoV-2 spike protein, and our spike negative EVs, which are just EVs collected from mock transfected cells, we see that they have somewhat similar size distributions, and we're able to get concentrations from those. We also performed transmission electron microscopy on our spike negative and spike positive extracellular vesicles. And you can see that we have typical EV populations with the negative stain. And interestingly, we wanted to see if we could perhaps detect or actually visualize the spike glycoprotein on the surface of the EVs. And I'm going to blow up an image here. But after discussion with our EM core that we were working with, they did say that it possibly looked like there was something resembling spike here in some of these EVs. And I kind of just highlighted what we're seeing here as to give you a sense of like what could be spike, but we're not 100% sure. Moving forward with proteomic characterization to look for the presence of spike protein, we performed mass spectrometry on our extracellular vesicles. And what I have highlighted in green here are just typical proteins associated with EV preps. I also listed some proteins here that are considered contaminants to give you a sense of purity. And interestingly, we did detect the SARS-CoV-2 spike protein amongst our purified EV population by mass spectrometry. And when we actually mapped the peptides that we detected onto the protein itself, you can see that we detected one peptide from the S1 subunit, which is as you can see here, is the subunit that contains the ACE2 receptor binding domain. This is important because this receptor binding domain is the target of most neutralizing antibodies. So we did detect one peptide from that S1 region, and then we detected a lot more peptides from the S2 region of spike. which was interesting and kind of, I'll talk a little bit more about when I show some Western blotting data. But the big takeaway here is that we were able to detect the wild type spike protein on our purified extracellular vesicle population. Now, when we did immunoblotting for spike protein, we used a C-terminal targeted antibody, which should be able to detect both full-length spike as well as the solitary S2 subunit. And it is known that in some cases the S1 subunit of spike can be shed, leaving only the S2 behind. And what you can see here, we did have to overexpose this blot, but what you can see is that there is quite a bit of the S2 subunit of spike present on our extracellular vesicles. And it's a little faint and a little hard to make out, but there is a very faint band there for the full-length spike protein. And interestingly, this data does kind of fit in with what we saw via mass spectrometry, which is that it seems like the S2 subunit of spike is much more abundant in the extracellular vesicles, and the majority of the spike we're seeing incorporated seems to be in the S2 form. But there is some full-length spike, at least according to this immunoblot. And that's kind of similar to what we saw with mass spectrometry. We also performed calnexin blots, which is an EV negative marker, which should only show up in the cellular lysates. So you can see that they are quite clean by calnexin. And we also did a CD63 Western blot in order to verify that we're, again, looking at extracellular vesicles here. And you can see that our EVs do have, both spike negative and spike positive do have CD63 present. So now that we've done our extracellular vesicle characterizations, we wanted to start looking at the functional assays, looking to see if these spike-containing extracellular vesicles can potentially bind to neutralizing antibodies and reduce their efficacy in blocking spike-mediated viral entry. In order to do that, we utilized a spike lentivirus pseudotype and an entry and fusion assay. Now this lentiviral pseudotype, it's basically, we used the SARS-CoV-2 spike D614G variant, which is a variant of spike protein that is incorporated better onto lentiviral particles. And our pseudotype system, it has the spike protein on the outside, but it has an HIV-1 core containing the protein beta-lactamase inside, which is relevant for our assay. So the way our assay works is we load our target cells with a fluorescent substrate called CCF2AM. This dye consists of two fluorescent moieties that are linked by a beta-lactam ring. So these readily enter the cell where they become membrane impermeable through the action of cytoplasmic esterases. And these two fluorescent moieties form a fret pair. However, if they're exposed to beta-lactamase protein, this fret pair gets cleaved and we get a differential readout via flow cytometry. Now, our spike lentiviral pseudotypes can bind to their receptor ACE2, get internalized, and then after membrane fusion, they release beta-lactamase into the cytosol, which then cleaves our dye. So we can basically look at spike-mediated, a surrogate of spike-mediated viral infection this way. So for our initial assays, just to characterize our spike lentiviral pseudotypes, or what I call SLVs, we basically take these lentiviruses and infect ACE2 293 T cells. Now, these HEC293Ts we transiently transfected with ACE2 at the time. So what you can see here, when we just have non-transfected HEC293Ts and we add our spike lentiviral pseudotypes, we do not see levels of fusion above the background of the assay. There's always a little bit of background here. However, if we transfect the HEC293 T cells with ACE2, we see an increase in fusion, or what some people would call viral entry, in a spike-mediated fashion. And just down here, I've shown that the transiently transfected HEC293 T cells do express ACE2 by flow cytometry. Now that we've shown that we can basically have a surrogate of spike-mediated viral infection, we wanted to make sure that we could neutralize that infection with a commercial neutralizing antibody. So we obtained an antibody from a company and basically tested it to see if it could block this viral entry or this viral fusion that we're seeing. And what you can see here on the left, we normalized our entry levels to the spike lentivirus by itself. When we add an isotype control antibody, we do not see a significant reduction in viral entry. And when we add this commercial neutralizing antibody designed to target the spike protein, we see a significant reduction in the levels of spike-mediated viral entry. And we tested this at just one dose here. But we wanted to look at this commercial neutralizing antibody across a broad range of doses in order to not only show that it generates a nice dose curve, but also pick doses to use going forward with our EV assays. And what you can see here is that the commercial neutralizing antibody does neutralize this lentivirus in a dose-dependent manner, and that what we call neutralization inversely correlates with fusion. As we get more neutralization, we see less fusion or less viral entry. Now that we've shown that we can perform neutralization assays with spike lentivirus, it was time to test whether these spike EVs could interfere with the ability of this neutralizing antibody to block viral entry. So for this EV interference assay, we mix first spike lentiviruses with particle ratios of either our spike or mock transfected spike negative EVs. And as I mentioned, we basically determined particle counts prior to this assay, and that's how we determined how many extracellular vesicles to add based on the particle count of the spike lentivirus. And we added it at 1:1 and 1:2, 2:1 ratios, sorry, of EVs to lentivirus. And then we added also our commercial neutralizing antibody. We mixed that all together before we then did our entry assays on the cells. And our hypothesis was that in the presence of these spike positive EVs, we would see decreased levels of spike lentiviral neutralization and the percent neutralization at the IC50 value of the neutralizing antibody. Basically, in other words, we would be seeing higher levels of infection in the presence of these spike EVs, because our hypothesis was that they can serve as decoys binding these neutralizing antibodies. So what you see here is one of these experiments done with the mock EVs, or the spike negative EVs. And surprisingly, what we found was that the spike negative EVs increased our neutralization activity of the antibody and reduced levels of infection just compared to the lentivirus alone without any EVs present, which was an unexpected finding. However, when we added the spike containing extracellular vesicle population, we saw the opposite effect. We saw that neutralization actually decreased, which means that infection increased, and that was really interesting. But these are just two representative images. So we performed this experiment multiple times, and we aggregated the data in this figure here. And what you can see when we've done multiple biological replicates of this is that these spike containing extracellular vesicles do reduce the neutralization activity of this neutralizing antibody when compared to the spike negative EV control. And it happens in what appears to be a dose dependent manner, where when we've increased the ratio of extracellular vesicles that we're adding, we see a even more drastic reduction in the levels of neutralization that's occurring under that antibody. And so that was really interesting to us, and it was also even more striking considering that the spike negative EVs had the opposite effect. So just the presence of spike on the surface of those EVs presumably led to a large decrease in the neutralization capacity of that antibody. And that's kind of what I'm summarizing here in this page, is that our spike negative EVs surprisingly increased the efficacy of the commercial neutralizing antibody, which also means that the spike negative EVs reduced infection by those spike lentivirus in the presence of commercial neutralizing antibody. And on the other hand, our spike containing EVs, our spike positive EVs, reduced efficacy of commercial neutralizing antibody, and they increased infection by these spike lentiviruses in the presence of commercial neutralizing antibody. Now, commercial neutralizing antibodies aren't necessarily representative of the normal or the real physiological situation. They're obviously made in the lab, and they might not be similar to what's happening in a patient that's actually infected with COVID-19. We asked the question, what about in a clinical setting, and what can we do to look at more naturally generated SARS-CoV-2 neutralizing antibodies? So in order to do this, we obtained patient serum from patients who were convalescent from COVID-19. So first here we obtained a COVID positive pooled serum sample, as well as a COVID negative pooled serum sample that was taken at the same time. And we wanted to test if we could perform neutralization with this patient serum. We also changed up our assay a little bit to accommodate the larger scale necessitated by performing serum experiments. So instead of using the spike lentivirus that delivers beta-lactamase to target cells, we used one that delivers an EGFP gene. So infected cells will produce EGFP. We also, at this point, obtained a stable ACE2 293T cell line. This cell line also expresses another fluorescent molecule, MIRFP670, under the same promoter as ACE2, and allows us to basically gate on only ACE2 containing cells when we do our readouts. So we tested this COVID positive patient serum for its ability to neutralize our spike lentiviruses and normalize that to the COVID negative serum pool. And what you can see here is that our COVID positive serum pool was able to neutralize our lentivirus in a dose dependent manner, as you would expect. And from this, we wanted to pick a few different dilutions of serum to move forward with, because from this point on, we're going to start collecting some individual patient serum samples. So this kind of gets to the crux of our study and the big figure, which is we're going to test to see if these spike EVs have the same effects on patient serum as they did on the commercial neutralizing antibody. Namely, can they reduce the efficacy of patient serum's ability to neutralize a spike-mediated viral infection? So for this, we obtained 9 different convalescent COVID-19 patient samples. Similar to before, we mixed the spike lentivirus with the extracellular vesicles and the different dilutions of patient serum we picked out from the pooled serum experiment. And then we infected these stable ACE2 293T cells and read out GFP 3 days post-infection. And at the end of the experiment, we normalized our infection levels to a no serum, no EVs infection control. And then we compare the effects of spike EVs to the effects of non-spike containing EVs. And our hypothesis was that the presence of these spike containing EVs would lead to decreased levels of spike lentivirus neutralization, decreased levels of serum efficacy, and increased levels of spike lentiviral infection when we compare that to the spike negative EV condition. So what you can see here is the data from these this big experiment, and we have all 9 of our patients represented here. And I've arbitrarily grouped them into 3 different categories of neutralization ability. So there's low neutralizers, there's moderate neutralizers, and there's high neutralizers. Now, our low neutralizers typically have curves that look to be at the top of a typical dose curve. They don't have a lot of neutralizing ability. And interestingly, what we see here with these low neutralizers is that the presence of these spike-containing EVs did increase infection levels, and by nature decrease neutralization levels to pretty significant levels when compared to the spike-negative EV condition. And that was really interesting, and it seemed like they had pretty drastic effects. In the moderate neutralizing patients, which had middle ground neutralizing capacity, you can see that again, the spike EVs did seem to increase infection and decrease neutralization ability of the patient serum across the different dilutions. And of course, we have our high neutralizers, which are those who can very strongly neutralize the virus. And what's interesting here is that with very little serum dilution with these high neutralizers, we don't tend to see differences in the effects caused by the EVs. And we believe that's because if you do have some sufficient levels of neutralizing antibody, you could overwhelm any effect that the extracellular vesicles might have by serving as decoys, because you'll just have enough antibody to bind to all the EVs and to bind to all the lentiviruses. So we kind of predicted that we wouldn't see a change in the we wouldn't see a change between the EV conditions at these lower serum dilutions of the high neutralizer patients. But interestingly, as we dilute their serum out, we do start to see some changes between the spike positive EVs and the spike negative EV conditions. Now, it's a little hard to take all this in from just looking at 9 different plots, so we wanted to aggregate this data to really show the EV decoy effect that I'm talking about. So just something important, I want to explain what the EV decoy effect is and what I'm referring to. So to quantify what we call the EV decoy effect, which is the ability of these EVs to influence neutralization levels, we calculated it by taking the mean infection in the presence of the spike positive EVs, and we subtracted from that the mean infection in the presence of the spike negative EVs. Basically, here we're subtracting the red point from the corresponding blue point, and then we're representing that difference on the y-axis. If this EV decoy effect value is greater than 0, that would mean that spike EVs led to an increase in infection and a decrease in neutralization. If that effect is less than 0, then spike EVs led to a decrease in infection and increase in neutralization. And lastly, if our EV decoy effect is around 0, that would mean that spike EVs had no differential effect than the spike negative EVs, which is basically the null hypothesis that EVs don't have any effect. Now, when we plot all these patients' serum dilution samples at this on this plot here, we have the EV decoy effect on the y-axis. We also have the percent of max infection, which is a measure of basically of neutralization in a sense. It's the inverse of neutralization. On the x-axis, this is a little bit more of an unbiased way of grouping these different data points into neutralization categories. You know, I did it arbitrarily prior to this, but here they fall where they do. And one patient might have 2 points that end up in the high neutralizing ability and 2 that end up in moderate. But I think the big takeaway here is that we are seeing a pattern of this EV decoy effect depending on the neutralization ability of that serum. It seems like the serum that has high neutralizing capacity, we don't see large effects of the EVs. In the moderate category, we see increasing effects, and in the low neutralizing patients, we see the highest differences caused by spike EVs. And interestingly, the general trend here is that almost all of the data points are above the 0 line, suggesting that across the board, spike containing extracellular vesicles did increase infection and reduce neutralization when compared to the spike negative EVs. And we did Pearson correlation to draw a correlation line and to get a p-value for this correlation. So there is a correlation between how strong of an effect do the spike EVs have on reducing serum efficacy, and how good was that serum at reducing infection in the first place. And it makes sense that if your serum it doesn't have a lot of neutralizing antibodies to begin with, that the EV decoy effect is going to be outsized because there's just less antibody to go around. And if a lot of it's binding to the EVs, then there's not a lot of it around to bind to the lentivirus. And this is just the same data, but grouped by the unbiased neutralization categories, and then displaying the basically the means or comparing the different means of the different conditions. And this shows the same trend that I mentioned already, which is that we see higher levels of the spike EV decoy effect with low neutralizing capacity of the serum. Now, we reached out to our collaborator that provided us the serum samples in order to gauge the antibody levels of these different patients. And what we found was very interesting. When we plotted here the neutralization ability on the y-axis, and down here are arbitrary units representing antibody levels. Our collaborator did provide us both spike levels of spike antibodies and levels of antibodies specifically targeted to the receptor binding domain of spike. And interestingly, our neutralizing capacity that we saw in our assay correlated very strongly with the antibody levels in those patients for both the spike antibodies and the specific RBD antibodies. And this tells us that our assay is a good measure of neutralization and can predict neutralization ability of these patients. So to wrap up with our conclusions, we found that these SARS-CoV-2 spike expressing cells can release EVs that incorporate spike. Approximately 80% of those EVs are CD81 positive and another 8% are CD9 positive, and they're not mutually exclusive. The majority of our EV-borne spike is appearing to be in the S2 subunit, which is interesting given that we saw effects on the serum neutralizing capacity. Our spike positive EVs increased spike mediated viral infection by reducing neutralizing activity of the commercial neutralizing antibodies and convalescent patient serum. And lastly, the spike positive EV decoy effect is stronger with moderate to low amounts of neutralizing antibody and seems to be overwhelmed by high amounts of neutralizing antibody. So for some of our future directions, we want one thing that we really think would be interesting to find out is what determines spike incorporation onto EVs. Basically, where does the pathway of spike production cross with the EV production pathway, and how does spike get on the surface? And moreover, what leads to different types of spike processing on the EVs? As we showed with our mass spec data and our Western blotting data, why is most of the spike in its S2 form and seems to have shed the S1 subunit, and what could lead to that? Another interesting thing would be to look at the spike EV cargo and the effect it has on recipient cells, namely what is in these spike containing EVs, and is it different than the rest of the EVs? Because it could have interesting implications for viral infection. And a connected point is this spike protein on extracellular vesicles capable of mediating EV cell membrane fusion? A lot of my past work has looked at how these viral proteins on the surface of EVs can lead to EV cell membrane fusion. So I'd be interested to know if it happens with spike protein, and it could also be relevant if there's different cargo in those spike EVs that is then being able to be delivered to cell cytosol. And this is something our lab's currently looking into. Another thread would be to look and see if we can detect spike EVs in the plasma of COVID-19 patients. And lastly, given that we've basically shown here for the first time that extracellular vesicles can serve as decoys for the neutralizing antibodies in SARS-CoV-2, do other viruses have similar patterns of EV-mediated and decoy activity? Can EVs from a variety of different virally infected cells serve as these decoys? And are viruses basically just utilizing the natural mechanisms of EV production of the cell to their advantage, which wouldn't be wholly unsurprising because we know that's what viruses do. So with that, I'd like to thank my lab, all the people who've helped me assemble this work, of course, many collaborators at Case Western and at the Cleveland Clinic, my thesis committee, and of course, I'd like to thank, again, Dr. Whitwer for allowing me to give this talk today. And I'm willing to take any questions.
Well, thank you very much, Zach, for that talk. I think this is some exciting work, and it has provoked some questions from our audience as well. So I look forward to the discussion now. And so I am going to allow participants now to unmute themselves, and I'm going to just go through the chat box. So if you have a question that you haven't asked yet, please use the chat box to pose that question or to make that comment. And I will go through here and ask you to unmute yourself. So if you've asked a question, get ready with your microphone to join the discussion here. So let's start out with a question about cell types and tetraspanin expression. This is from Melissa Guadron Lopez. Can you go ahead and unmute yourself and ask Zach your question?
Yeah, hello. Can you hear me?
Yes.
Yes, loud and clear.
Okay, good. Thank you very much, Zach. It was very interesting, very nice work. I was wondering about this proportion of spike EVs and the tetraspanin. So in your opinion, it is this proportion, CD80 positive, more about co-expressing spike EVs related to these particular cells? I mean, the proportion of tetraspanning of the HEC cells. It is this conserved in another cells?
Sure. I would predict that it would probably vary by the cell type. Of course, we saw that only with the HEC. We only did this experiment with the HEC293T cells, which in that case we saw that the majority of them were CD81 positive. But I'm not sure. I wouldn't go as far to say that that would hold the same for every cell line we tested. It was helpful for us in that we expressed CD81 to verify that it was extracellular vesicles, at least hard carrying extracellular vesicles that were carrying the spike protein. But I would expect that it could probably vary quite significantly based on the cell line that we're using at the time, if it was a different cell line.
Okay. Thank you very much.
Thanks for the question. And our next question is also having to do with the production from the cells. And I love this because I love stoichiometry. So Ting Ting Shao, could you ask your question, please?
Hi, thank you for this great presentation. My question for you is maybe I missed your data, but I want to know how many cells you used to secrete these transfected cells to collect the EVs, and how many EVs you detected with the spike proteins. I mean, is this very significant to look at because they act as decoy? I don't know if I'm making myself clear.
No, I understand. So in terms of how many cells, you're gonna make me do a little bit of math here, because we do, for our purified EV populations, we typically harvest them from about 12 different 10 centimeter dishes, which each one has it's about 30 million, approximately 30 million HEC293T cells, if I'm doing the math right. 2.5 million per dish, and then 12 dishes, and then we transfect the cells. And in terms of how many of the EVs have spike, our data suggests that it's not a whole lot of them. It's kind of rare. It's probably under 10% of the released EVs from those cells have spike. And as you saw from my mass spec and Western blotting data, that doesn't necessarily mean that those EVs all have full-length spike. There's probably even less of a fraction that have the full-length spike, which is kind of interesting. But I wouldn't say it's a whole lot, but they're definitely present. I mean, we were able to detect with the nanoscale flow quite readily, and the Western blotting, we were able to see it too. The S2 brand was actually quite bright. So I don't know. Does that answer your question?
Yeah, I think it answered my question, but it's I ask this question is because in the real time, are these EVs really act as act to protect this virus from these antibodies? I mean, if there is not very much of them with this antigen, so maybe it's just secreted by the EVs. And also these EVs with the spike protein, are they secreted from the virus, or they are secreted from these transfected cells? And how
So is the question, are you asking in a clinical situation where there's actually infected cells in a patient, are you asking, are those cells secreting spike containing EVs, and at what level? Is that your question, if I'm understanding?
Yeah, because you have, if I understand it right, it's you have this this experiment based on the hypothesis in the very beginning, that you see this virus with this this extracellular vesicles with this with a spike protein, they can act as decoy to protect this virus from these antibodies. So I mean, if in your experiment in vital transfected cells, if they're not very much EVs with the spike, but would this mean in the real time they are not very significant to act in this way? I
Well,
mean,
well, I'll say we don't know to what level they would be in, you know, in the body if someone was infected. Of course, all these experiments are just done in vitro with cell culture, so it's hard to necessarily connect that to the ratio of spike EVs that are released by cells compared to actual lentivirus or sorry, actual SARS-CoV-2 virions in an actual infected cell. Yeah, I guess it's obviously a different situation, but even though we did see, you know, that the presence of spike wasn't on all EVs, but clearly there was enough there that it did have a significant effect on the ability of the serum. So I guess I don't know if I can answer further.
thank you. I think you answered me my first question. Thank you.
Yeah.
All right. So next we have Swathi Balaji, and I think that Swathi has asked several questions here. So Swathi, go ahead and get started, and then we can maybe have a back and forth between you and Zach, and then we'll bring some other people into this too. I think some very interesting questions here.
Thank you, Ken. Zach, thank you for the fantastic talk. You know, I had similar questions as the previous speaker. You know, first of all, in the patient sera, do we see there is a correlation or association between the number of EVs with spike proteins versus the viral virions? And then my other question was, you know, what do you think is the mechanism at action? Like, when you have these EVs with spike proteins, is it just changing the stoichiometry of these neutralizing antibodies by being present, or do they have a more affinity or some kind of a preferential binding to the neutralizing antibody than the actual viral particles?
Right. So could you repeat your first part of the question again?
Do you know in patients that have different levels of infection or response, you know, with the COVID patients, just like any other disease, some people are able to come back home and some people, you know, we lose them forever. Do we know in those specific patients, you know, if you divide them into low, medium, high susceptibility patients, do you know if there is a different level of these EVs with spike proteins in those patients? In their convalescent sera, how is it changing? And the follow-up question is also, when you take the convalescent sera from these patients, is the timing very similar, you know, since they had their initial infection, or was it just from different time points as it became available?
So first to answer the first part of the question, we didn't actually detect extracellular vesicles with spike in the convalescent serum. Maybe I'm misunderstanding, but we never looked for these spike EVs in actual like patient blood or patient serum. So it's hard. I guess I'm not sure if I can answer the first part, but as for the serum classifications, we obtained the serum from a collaborator. So I know that these are convalescent patients, so they've all recovered from the disease. So I'm not sure I guess I'm not sure I can answer necessarily what you're asking.
No, I think this is a great study scientifically, but I think, you know, to prove that it has relevance in the disease pathogenesis, I think, you know, just now, you know, working a little bit more from a clinical standpoint of understanding what happens to these EVs during active infection and during the convalescence phase.
Sure.
I think that will really elevate the overall impact of the science.
Yeah, I think you're definitely right. Like we would be interested at some point in, you know, actually looking and finding these EVs in actual, you know, COVID patients and people who are infected, and I'm sure there'd be something interesting there with how many spike EVs that they have and how their clinical course goes and how well their body responds and how well the neutralizing antibodies respond. Because I know there's been research that's shown that often these neutralizing antibody responses against actual COVID-19 infection are not usually too good. So I'm wondering if, you know, there could be some correlation there with the levels of spike EVs that are present. And to answer the stoichiometry question, my guess would be that it's just by virtue of the extracellular vesicles being present and being capable of being bound by these neutralizing antibodies, it just reduces the number of available antibodies to bind the lentivirus. But, you it's possible that there could be something else going on, that somehow maybe those neutralizing antibodies bind even more strongly to the EVs than they do the normal viruses. And that'd be something interesting to look at, because that HIV paper that I cited at the beginning of my talk, they actually found that the immature HIV particles did bind the neutralizing antibodies much more strongly than the mature infectious particles. Yeah. So that, that kind of makes sense that they're being used as a decoy, because they bind the antibodies even better. And it's possible that these EVs bind antibody better than the SARS-CoV-2 virions. But that would be work we would have to look into.
Thank you so much, Zach. Good luck.
Thank you.
We've had several comments and questions actually along these same lines from Phil Askenase, Glenn McConkey, Lee Sun. So any of you, if you would like to unmute and continue this discussion, feel free.
Yeah, Phil Askenase, there's a rather emergency thing here. The journal you submitted to, probably if it's worth its value, won't accept this paper unless you show that there are such decoy vesicles in the real life. And it's a very important point, because convalescent plasma has been very, very disappointing in these patients. And I've been pushing for inhibitory effects of the exosomes that's in the convalescent plasma. So this should be your number 1, 2, and 3 thing to do, to see if the convalescent plasma has exosomes or EVs that have this blocking effect.
Yes, that would be definitely something we would like to look into. And I do wonder though, if a person in convalescence not actually having active infection, would any spike containing vesicles at that point be cleared by the immune?
You don't know until you look.
That's true. That's true.
And right now we know that convalescent plasma doesn't work well.
Yeah.
I think there are EVs that have functional consequences on antigen presenting cells and T cells. Mm-hmm. But you don't know until you look.
Sure.
And you're in a very good position to look.
Yeah, so very interesting to find out if any of these antigens remain, after acute infection.
My guide in working with EVs is don't make assumptions.
Sure.
Because they're a different world than the world you come from which you make assumptions. Let them teach us the truth. Don't take previous truths as assumptions.
So speaking of,
get to work on this right away.
Of assumptions, yes, that's a good point, Phil. And so and teaching too. So Michael Grainer has a comment here coming from the tumor antigen world. Michael, would you like to share with us the experience that you had with the tumor antigens?
Sure, thanks, Ken. So this is exciting to those of us in the tumor world to some extent, perhaps, because we proposed for a long time that vesicles in the bloodstream of patients likely contain tumor antigens, and if you're generating antibodies against those, those can be titrated out simply because they're floating by, far more likely to encounter the antibody there than they are near the tumor. And this has actually been demonstrated at least in vitro with transmutamabs, Herceptin. So for us, and then in general, this has been met with an extraordinary amount of unconcern. So I just wanted to comment that I thank you for bringing this to the forefront of science again. And you've done some very excellent techniques here to come up with, I think, some really good displays of how we can study these things. And yeah, going back to Phil's comments, you need to find that in the vesicles in patient sera as well. And as well as there's going to be a myriad of immune downstream plays that those vesicles are going to cause on the immune response outside of just grabbing antibody, which is, I think, extremely important. But I just want to thank you for what you did with that. And one quick question, which was in there somewhere, I may have missed this. Were you using commercially available neutralizing antibodies?
Yeah.
Those were polyclonal or monoclonal?
So, oh geez, I'll have to look back. If you can email me and I can tell you about the commercial neutralizing antibody. I know it came from a company called Xenobiological.
Oh, okay.
I don't recall offhand if it was polyclonal or I think it was monoclonal, but I can't say with definitive. So,
well, I mean, because we're using Regeneron antibodies, which are monoclonals,
sure.
in the clinic. but the convalescent sera is very likely to be obviously polyclonal,
Right.
and also capable of hitting other antigens on the viruses as well. So just that's another complicating factor to make your life harder. So I'll wrap up with this. Thanks.
That's definitely part of the rationale for why we used patient serum, was because obviously the monoclonal antibody is one thing, but being able to show the same effects of the spike EVs in the context of many different antibodies against, you know, multiple antigens of spike even, I think that was really kind of the more physiologically relevant thing to look at. So thank you.
Now the next question, and actually a couple of questions from Artem Jivalajny. Would you like to ask your question about the EVs and why they might be there, what they might be doing with the immune system?
Yeah, general, my question is about evolutionary purpose of this spike EVs mechanism. My question is this mechanism for host defense? Is it the host defense system? Because there's multiple data that vesicles can somehow stimulate immune system, or is it a virus protection mechanism to defend himself?
Yeah, I think the way that we framed it here is that it's somewhat of a viral defense where you know, viruses already are known to hijack cellular systems for their benefit, and EVs are just another system of that cells have that can be hijacked. The virus can hijack the system of EVs to produce these decoy particles, and that allows them to escape detection by the immune system. So I kind of frame this in a pro-viral light, but you're certainly right that EVs from virally infected cells have been shown to have both pro-viral and antiviral functions. So it definitely required more elucidation, I would say.
Can I ask a technical question? This is Swati Balaji again. When you chose your filtration system, did you choose that specifically to preserve the surfactant of the EVs versus the cargoes? And, you know, did you try other techniques? You know, because nowadays we hear so much about, you know, a kit-related effect. How do you, or, you know, something that is very technically, your results are a product of the technique that you used.
Yes.
Have you tried this with a different vesicle isolation strategy?
Thanks for the question. You're right that we picked tangential flow filtration because we were avoiding ultracentrifugation techniques because of the damage that they're known to cause to EVs and the surface proteins. And especially envelope glycoproteins of viruses are typically not super stable under ultracentrifugation conditions. It of course depends on the viral envelope protein you're talking about, but with SARS-CoV-2, we kind of went straight to using a different method like tangential flow filtration that's much gentler on the EVs in terms of the forces they experience. And it also allowed us to, it allows you to concentrate them to much higher levels than other techniques, which was important for our assay. But you're right, we didn't try these same experiments with, let's say, ultracentrifuged collected EVs, or those had been put through ISON QEV columns or anything like that. And it would be interesting, you know, maybe I guess it's possible that this could be an effect of the technique we used to produce the EVs.
Okay, well, so we have some additional questions in the chat box here, but I'm afraid we're going to have to end here for now. And I will share these questions with Zach so that he can follow up with you individually. So I see that E. Gonzalez, S. Srivastava, Prussian Data, and some others have some questions. So I'm going to just share those. We'll make sure to get in contact with you about your questions. So Zach, thank you so much again for this presentation. Thank you for doing this work. I wish you all the best as it continues. And thank everybody for joining today. I hope to see you again very soon in an EV club. Take care, everyone. Bye now.